Good morning, everyone. Afternoon, evening, depending upon where you are. I am thrilled that you've all been able to join us. I don't know how many of you there are yet because I haven't been playing with my team system. For those of you who don't know me, I'm John Glass. I'm here at the J. Craig Venter Institute. We've got today is I'm happy to say this is our fourth edition of the Minimal Cell Workshop. Over our next three sessions, I think we're going to have some terrific talks. I will start out. I'm going to introduce things. Let's see. I'm going to share my screen if I'm remembering how to do this. So it's our fourth workshop, and I'm thrilled that we've got so many of you here. I like this beginning I used last year, so I'm using it again. So happy that everyone is here. I like to start off. I want to just offer you a few images of our cell, different things you can see. This is this thing called CellWork, an immersive biology education tool for Apple Vision Pro by the Canadian web developer Tim Davison. It's a slick tool that he's got there. There's a ribosome that you see inside the cell, or a pyruvate dehydrogenase complex. We've got, of course, this image done by Tom Dernick at the and Mark Ellisman from the UCSD National Center for Imaging and Microscopy Research. There's hundreds of cells crammed together. We've got some images from Ji-Woon Kwon and TJ Ha from now Harvard, where we're seeing cells. So you can see these cells at this super resolution microscope at the University of Illinois at Champaign-Urbana. Makoto Miyata's group has made this dazzling image of JCBI SYN3B, where they added 5 genes to make the cell swim around. I just love this image. And I like to think that I would have been courageous enough to try this experiment, because I was not expecting this to work when I first heard about it. But there it is. Some negative stains of the minimal cell. A fried egg colony for you mycoplasma fans. The cryotomography from Elizabeth Villa's group at UCSD. On the right, you can see ribosomes, and we average out to about 700 per cell. A computational rendering of the minimal cell done by Zane Thornburg from Zan-Luthi Schulten's group at the University of Illinois. This thing that James Pelletier and Elizabeth Strahowski did, and it's a dazzling image of JCBI SYN 3.0 inside a microfluidic chamber, and you can see how just aberrantly wrong when you think about cell division this is. Of course, this image that I'd love to have a tattoo of, only actual size, I think, the classic minimal cell image, again, from Tom Dernick and Mark Ellisman at UCSD. We keep getting more publications. They're up to 70 publications now that mention in one way or another JCBI SYN 3, 3A, 3B, JCBI minimal cell, SYN 1.0, these sorts of things. So we're really coming along. And again, this idea that if you build it, they will come. And so many of you who are here today are part of this movement that we've got going with the minimal cell, where we built the thing and so many of you are using it. We are now up to, and I haven't updated this slide, it's now 80 labs that are using it as opposed to 70. And so I need to be putting new dots and updating this slide. But I'm just really thrilled with this, the way this is all going. Again, we're trying to use this tool to investigate the first principles of cellular life. Our vision is that if we have a cell where every gene is needed and we know the function and biological role of every gene product, we may be able to develop algorithms for genome scale redesign we may be able to understand cells better than we ever have. And then there is also the possibility that we may be able to use our minimal cell for applied purposes. My colleagues at the JCVI, including Daniela Bittencourt, published a paper last year, or this year actually, where we're using JCVI SYN3B to investigate the pathogenic potential of proteins expressed by intractable pathogens, but we might also be able to use the minimal cell perhaps as an anti-cancer therapeutic or for some other things that are really cool. So I think that there are just lots of things that we can do with this cell, and I'm so looking forward to hearing what all of you who are assembled today, what you've got. If you would still, if you haven't submitted a talk and at the end of the day you decide you would like to talk, feel free to send me an abstract, a title, and I will get you added to the schedule for Thursday or next Wednesday. Some housekeeping things. Tim Stayes is online. If you should need him or have issues, you can contact him via chat or email, and I'll be monitoring the time. But basically, each of you have 15 minutes to talk and do questions.